Well, thank you, Ann. It's a real pleasure to be here and to be able to talk about some of the work that's going on in the area of molecular cytogenetics these days. I'm going to try to give a general overview of the whole field, but I'm going to illustrate this using work from the division at San Francisco. And so to start, I'd like to acknowledge the fact that I've borrowed work from my own lab, from Dan Pinkel, Fred Waldman, Kallioiniemi, and Masaru Sakamoto. Well, what I'm going to try to do is to first start by discussing some of the technology associated with molecular cytogenetics, specifically fluorescence in situ hybridization. This is basically a technology that allows you to look inside individual cells and chromosomes, and instead of seeing the somewhat featureless domain that you normally see in the light microscope, fluorescence in situ hybridization allows you to look with almost molecular precision at the organization of DNA sequences inside the interphase nuclei and in the mitotic chromosomes. And it turns out that this technology is important in a variety of applications these days. Depending on the probes that you choose, it allows you to investigate the nucleic acid organization of the interphase nucleus. In terms of the Human Genome Project, it allows you to localize cloned DNA sequences along a particular chromosome. And I think probably most importantly, allows you to begin to have a look on a cell-by-cell basis at the genetic aberrations that are associated with genetic diseases. Now, I'm not going to talk much about chromosomal organization of the nucleus and the physical mapping because these topics are going to be covered later in this meeting by people like Barb Trask and Jeannie Lawrence. Instead, I'm going to start with a discussion and overview of the technology and then talk about its application in the characterization of several genetic diseases ranging from things like Down syndrome prenatally to the diagnosis and prognostication in human solid tumors. So let me start with the general technology. What we're trying to do, as I mentioned earlier, is to stain specific regions of the genome using fluorescence in situ hybridization. And the approach here is to start by denaturing the DNA in the target cells or chromosomes so that it becomes single-stranded, and to react that with a suitably labeled nucleic acid sequence that is homologous to the region of the genome that you'd like to stain. Now, there are a variety of ways of labeling the probe so that you can render it visible in a light microscope later. It might be biotinylated, might be treated with the carcinogen acetylaminofluorine, labeled with the deoxygenin, and more recently it's possible to hang fluorescent molecules directly on the DNA probe. In any case, once you've got the probe labeled, then you can subsequently detect that with a fluorescent reagent. Now, the idea is to mix the probe and the target together and allow the single-stranded sequences to re-anneal under conditions where you have binding the probe only to DNA sequences to which it has high homology. Now, it turns out if you're working with human chromosomes, they're full of interspersed repeats that are not chromosome-specific, and so these are usually competitively inhibited during the hybridization process. Once this reaction has gone to completion, then you can detect the hybridized probe with a fluorescent molecule, and we often use molecules like fluorescein or Texas red so that they fluoresce yellow and red, and counterstain the remaining DNA with a DNA-specific dye like propydiamidide or DAPI. Might point out that since we have all of these different detection reagents, it's possible not only to detect one probe, but to detect several. You might label one yellow, one red, one yellow and red, and so on, so that you can look at multiple genetic events simultaneously. Now, the key to this whole technology is to have cytogenetically informative probes. And this part of the technology is rather well developed today. I'll talk about three classes of probes that are particularly useful. The so-called repeat sequence probes, which stain regions of the chromosome centromeres. This is particularly useful for counting chromosome copy number. Whole chromosome probes, which allow you to stain entire chromosomes, and specific locus probes that allow you to detect individual genes or segments of the genome. This is an example of one of these so-called repeat sequence probes. As I mentioned, these are probes that typically bind to the chromosome centromeres, and they target DNA sequences that are tandemly repeated over and over again, so that you may have several hundred thousand base pairs to several megabases with a target DNA sequence that can be detected using a probe that's only a few hundred base pairs long. The advantage is that these things are tightly localized. They can be made chromosome-specific, and they're intensely fluorescent, so that it's easy when you're looking at the metaphase spread to see which chromosome you're dealing with. And more importantly, if you're interested in looking at the interphase nucleus, you can also see the localization of the sequences targeted by this probe there as well. And so this turns out to be useful if you're interested in trying to investigate the organization of the interphase nucleus. And if you're interested in studying genetic diseases such as cancer, it's also possible to use these probes to do things like detect chromosome copy number, which is illustrated in this slide. Here you can see a crude metaphase spread, which is about all you get in a solid tumor, that shows 3 copies of the probe here. Here you can see an interphase nucleus that shows 4 copies of the probe. So it gives you a quick way of doing chromosome enumeration using this technology. Now, another probe that is commonly used these days are the so-called whole chromosome probes. These are probes that are composed of a large number of different individual DNA sequences that are combined together from a single chromosome type, so that you can use these as a composite staining reagent. And here you can see hybridization with one of these whole chromosome probes for chromosome 4, staining almost completely the 2 copies of the number 4 chromosome in this normal human metaphase spread. The way these stains are made builds on the fact, or derives from the fact that we've learned almost 2 decades ago now that it's possible to use fluorescence activated cell sorters to purify chromosomes of a single type. And so that it was then possible to take chromosomal DNA of one type and cut it up into a large number of different pieces, clone it into recombinant DNA vectors, and then use this library of probes as your hybridization reagents. So in this way you can get a large number of different DNA sequences all from the same chromosome type. This allows you to see these sequences not only in metaphase, of course, but also in interphase. And again, we have a reagent that's useful for people who are interested in studying the chromosomal organization of the interphase nucleus. We had originally thought that this was going to be a particularly interesting field of study, hoping for some rather regular organization of these sequences in interphase, but so far it's turned out to be remarkably random. One main application of these stains, though, is in detecting induced genetic damage, for example, by radiation. And one of the common types of aberrations that radiation produces are these structural rearrangements where you break a couple of chromosomes and have rearrangements between the two to either form dicentric chromosomes or translocations. And one of the nice things that this technology allows you to do is to detect these kinds of aberrations very easily. In fact, just almost at a glance. The idea here is to stain some of the chromosomes using in situ hybridization, but not the others, so that you can see exchanges between stained and unstained chromosome types as these bicolor chromosomes. And it looks almost exactly like it does in this cartoon. Here you can see one of these bicolor chromosomes that was induced by ionizing radiation. You can see one part of the derivative stained chromosome here, and the other part located there. And so what this gives you then is a very nice tool for measuring aberration frequency, in this case shown as a function of dose. What this allows you to do in the case of radiation-induced genetic damage is to look at the lymphocytes of exposed individuals and to measure the aberration frequency, and from that to estimate dose. And it turns out that this technology is being used increasingly in studies of the large exposed populations, such as the Japanese A-bomb survivors or the Chernobyl victims. The advantage of it is that it allows you to see stable translocations, which remain years or decades after exposure, so it gives you a nice long-term dosimeter. The other application that I'd like to mention just in passing is in prenatal diagnosis. One of the most common events that's found prenatally is trisomy 21 associated with Down syndrome. Finding or scoring cells for trisomy 21 usually requires an experienced cytogeneticist. In this case, if you take a probe for chromosome 21 and hybridize to a metaphase spread, you can see the 3 number 21 chromosomes almost at a glance. And so that it makes the technology much simpler to apply. It increases its accuracy, and it increases the speed of analysis. And probably it will eventually set the stage for almost complete automation of the whole approach. And I might point out that I've talked now about two classes of probes, repeat sequence probes and whole chromosome probes. These things are now available for essentially all of the human chromosomes, and many of them, in fact, can be purchased commercially. So this technology is now moving to the point where you really don't need to have molecular biological background to apply it. You can essentially buy the reagents as stains, use them as off-the-shelf reagents, and move quickly into these kinds of analyses. These whole chromosome probes also allow you to look for aberrations in interphase nuclei. Here you can see hybridization with chromosome 21 to a patient with Down syndrome. And you can see the 3 copies of chromosome 21 in this nucleus and 3 copies of chromosome 21 in this nucleus. But you can see over here that in this normal spread, things are a little bit less clear. And in fact, if you score these normal and Down syndrome cells in a blind way, what you find is that only about 60 or 70% of the normal cells show the expected 2 domains, and only about half of the trisomic cells show the expected 3 domains. And the reason for this is that these produce fairly large hybridization signals. And so there's a reasonable probability that they will overlap and give you an incorrect reading. And so the way around that is really to move to these so-called locus-specific probes. Here, the idea is to take a probe, a small probe now, to a DNA sequence that you're interested in studying. What I'm showing here is an example of hybridization with a probe to the retinoblastoma gene on chromosome 13. Turns out that inactivation of both copies of this gene typically leads to retinoblastoma in children. Here you can see very clearly the presence of the retinoblastoma sequence or gene on chromosome 13 here in metaphase, and here in interphase. And so it's a straightforward assay to actually use these probes and then to do a quantitative analysis of probe copy number. Here you can see that if you look at the normal cells carrying both copies of the RB gene, look at the frequency of cells in the population showing 0, 1, 2, 3, 4 copies of the gene, what you can see is that almost all of the cells shows the expected 2 copies. On the other hand, if you look at cells that have lost one of these RB genes, you can see that the majority of cells in the population show only a single copy. So it is a good technique for doing copy number analysis. I might point out that using these kinds of probes, you can target DNA sequences that are as small as a kilobase, although the efficiency with which you can detect those probes or those sequences goes down as the probe gets smaller and smaller. Well, the other thing that you can detect using these kinds of probes are structural rearrangements. One of the genetic diseases that we know a lot about is chronic myelogenous leukemia. Turns out that this disease is almost uniformly associated with the rearrangement between the BCR gene located on chromosome 22 and the ABL gene located on chromosome 9. As a result of a translocation, these 2 genes are typically brought into close proximity to one another. And so using in situ hybridizations and detecting one probe, let's say in green, second probe in red, it's straightforward to look for the fusion of a red and a gene sequence as a result of this rearrangement. If you were to look in an interphase nucleus, what you would typically see for a normal cell are 2 red and 2 green spots that are more or less randomly distributed around. If you were to look in one of these leukemic cells, what you see very frequently is the fusion of one red domain and one green domain. And if you actually apply this technique using these probes, and you're sitting in the front row, what you can see here for these leukemic cells is that you have a red spot here, a green spot here, and a yellow or a red-green fusion here. This is the fusion, gene here, fusion here, red there, and so on. So this gives you a ready technique to pick up these rearrangements even when they occur at relatively low frequency, and this opens the way for studies such as detection of residual disease even when it's not clinically apparent. Now, the difficulty, as those of you in the back row can tell, is that these are difficult to see. You have to be fully dark adapted, and it takes a long time to score these things just looking through the microscope. And so one of the things that we've been working to do is to develop computer-assisted microscopy to try to facilitate the visualization and scoring of these events. And the general approach that we use is to use a conventional microscope equipped with two cameras, one of them a cooled CCD for high-resolution image acquisition, and the second an intensified CCD for roaming and focusing. Turns out that the cooled CCD is excellent for high-resolution analysis, but it takes about a second to read out one of these cameras, and so that's not particularly advantageous for roaming around on the slide looking for things to study. Now, once we have acquired an image that we want to analyze in detail, we then acquire a multicolor image using the CCD, transfer that image to the Sun system for archiving, and then subsequently to a different Sun system for offline processing. And this system is being developed or has already been developed for a variety of applications to improve display, for physical mapping of DNA sequences along chromosomes, to measure the ratios of different colors, to count spots in interphase nuclei and to measure their proximity, and to do things like look for rare events and automated analysis such as things such as translocations. I'm only going to talk about a few of these things today, but the rest of them are clearly well along. Well, the general approach here to using microscopy for fluorescence in situ hybridization is to acquire multicolor images with negligible registration shift. The idea here is that we change the filters in the microscope so that we generate a blue image on the CCD, a green image, and a red image, acquire these three images separately, and then combine them in the computer to give us our multicolor image. Now, since we're interested in such things as proximity, it's really important that we have negligible registration shift. And in most light microscopes that you buy commercially today, if you change filters in the optical pathway, you will move the position where the image comes out on the camera so that the green image, for example, is not in proper registration to the red. To overcome this problem, we worked with a variety of filter manufacturers to develop a multichroic beam splitter that goes in the optical pathway of these epi-illumination fluorescence microscopes. And these beam splitters have the characteristic that they transmit in the blue, green, and red, and reflect in the ultraviolet between the blue and the green and then the yellow range. And so simply by selecting an external filter, we can selectively excite dyes like DAPI, fluorescein, and Texas red, and acquire images of these on the cooled CCD without ever changing anything in the optical pathway. And we've shown that this gives us what we were striving for, namely registration-free multicolor image acquisition. And what this allows us to do then is to use the computer system to acquire and process images, giving us the information that we want. This is an example of some of the mapping software that we've developed for the system, which acquires images of metaphase spreads, locates the hybridization signals of DNA sequences along the chromosomes. In this case, this is a probe for the oncogene CMYC. The computer program also outlines the chromosomes, calculates its medial axis, and maps the chromosomes along the medial axis of the chromosome. The other thing the system allows you to do is to acquire images of relatively weakly fluorescing cells. This is an interphase nucleus stained with a repeat sequence for chromosome 8, and also a probe for the CMYC oncogene on chromosome 8. And if you focus on this spot down here for a moment, one of the things that I want to point out is that this spot has actually got two colors in it, a green spot and a red spot. What we're actually looking at here is the 5' end of the CMYC gene and the 3' end of the CMYC gene. So it gives you an idea of the sensitivity that you can generate with this kind of a system. Well, that's basically a summary of the technology for fluorescence in situ hybridization that I wanted to cover. What I want to do now is to spend the rest of my time talking about one application area for this technology to try to give you some feeling for its power. In particular, I want to focus on the molecular cytogenetics of human tumors. There's a variety of reasons for wanting to do this in our lab. We're certainly interested in prognostication, that is to say, a prediction of how the tumor is going to respond to therapy or how it's going to progress with time. We're interested in positional cloning of genes involved in the progression. Turns out that there are a large number of both oncogenes and tumor suppressor genes that are likely to be involved, especially in solid tumors, that have yet to be cloned, and we think that molecular cytogenetics has an important role in trying to facilitate the cloning of these genes. And then finally, we're interested in some of the basic biology of genetic evolution, and I'll talk a little bit about each of these areas in the remainder of this talk. Now, in terms of prognostication, there's a variety of endpoints that one might look at trying to determine how a tumor is going to behave in the future. Historically, people have looked at histologic grade, stage of the tumor, whether or not it's metastasized to the lymph nodes. People have looked at immunophenotype. The approach that we're taking is to try to develop a prognostication or a tumor grading scheme based on the genetics of the tumor. And there are three endpoints that we're considering for the development of this genetic grading system. One of these is genetic instability. It turns out that there are genes such as P53 now that have been identified that seem to play a controlling role or driving role in the development of genetic instability, and the idea is that the more unstable a tumor is, the more rapidly it will evolve through the collection of genetic events or aberrations that are needed to take it into a metastatic state. And so we would like to have a measure for genetic instability. Another possibility is that, sort of related to instability, is that tumors progress through the accumulation of a variety of genetic aberrations, and the idea is that the more you have, the more likely it is that the tumor will be well along in the evolutionary process and possibly will have metastasized. And finally, there are specific genetic aberrations that may do things like impart to the tumor increased proliferative rate, the ability to invade, or the ability to metastasize. And so we would like to use molecular cytogenetics to look at specific aberrations as well. Well, let me start with prognostication according to instability. I mentioned in the beginning of this talk that we had probes for repeat sequences that were chromosome specific. And this is an example of the application of one of those, or several of those probes, in the analysis of a primary breast cancer. This breast cancer, by all measures other than this, was diploid, that is to say it had a normal DNA content. And so we looked at chromosomes 1 and 7 and 11 and 15 and 17 and looked at the frequency of cells in the tumor that had 0, 1, 2, 3, 4, 5, 6, and 7 copies of these particular chromosomes. And what you can see in this tumor is that it's rather well behaved. What you can see is that almost all of the cells in the tumor have the expected 2 copies, although there are some cells that have extra copies of chromosome 1. But by and large, this is a well-behaved tumor, and so we would consider this not to be not particularly unstable. Therefore, it might have a good prognosis. This remains to be tested, but that's the hypothesis. On the other hand, if you look at this tumor, which is, again, a primary tumor of the same grade and stage, what you can see here is that it's wildly unstable. Some of the cells have 2 copies of these chromosomes, as you would expect, but others have 4, 6, 8, 10, 9. In fact, if you look out here, this is chromosome 17, you can see almost 20% of the cells in this cancer have 12 copies of chromosome 17. So what you can see here is that this is a much more heterogeneous, and therefore we postulate unstable tumor than the previous one, and we would suggest that this may have a worse prognosis. And we're waiting for the follow-up on these studies now to determine whether or not that hypothesis pans out. Well, another possibility that I mentioned is prognostication based on accumulated genetic aberrations. And there's really two classes of aberrations that one might look at in human solid tumors. The oncogenes, these are typically dominant genes that are activated either by amplification or overexpression or translocation. And if you look at breast cancer, you find that oncogenes such as ERBB2 and CMYC, there's a region on chromosome 20 that's amplified. These tend to be very frequently involved in the progression of breast cancers, and certainly amplification of ERBB2 has been prominently suggested as a poor prognostic indicator. And so what we would like to do is to ask the question, can we use this technology to look at these events? At the same time, there are tumor suppressor genes. These are typically recessive genes that are only activated if you lose the normal allele. So you might have one mutant allele, which is the mutant tumor suppressor gene, and you lose the normal allele, that activates the mutation. And genes that fall in this category are P53, NM23, RB, and it looks like that there may be tumor suppressor genes located on a number of chromosomes in breast cancer, 1P, 1Q, 3P, 11, 16, 17, and so on. And again, we are asking the question, can we use molecular cytogenetics to detect these aberrations? And the answer in both cases is yes. If you're focusing on gene amplification for a minute, again, this is the scheme that you would use to pick up the amplification of the oncogenes. The idea here is to use a probe to the oncogene that you want to detect, and a second probe to a different region of the same chromosome, either elsewhere on the chromosome or perhaps at the centromere. And if you were looking at a normal cell, you'd expect to see 2 copies of each gene. If you look at a cell that's carrying an amplification event, what you would expect to see is 2 mini copies of the probe that binds to the oncogene. And so the idea here is that the number of hybridization domains indicates the degree of amplification. And as I'll show, if you use conventional molecular studies such as Southern analysis, the results that you get out of this correlate very well with them. This is an example just showing the application of this. This is hybridization with 2 probes. One of the probes is to the centromere of chromosome 8. This is the chromosome that I mentioned earlier that CMYC, the oncogene CMYC maps to. The other probe that we're using here is to CMYC, and it's detected in green. What you can see here in this ovarian cancer is that there's relatively low-level amplification. You have 2 copies of chromosome 8 in most cells, and you have a half dozen, maybe a dozen copies of CMYC. And so this gives you information that says not only is the gene amplified, but it tells you how much. Now, they don't all look like this. You can also have cell lines where you get massive amplification. This is a colon carcinoma cell line in mitosis, and here you can see literally hundreds of copies of CMYC. There's so many that they tend to be run together and look like large blobs. And just to make the point, if this happens to be a study of a different oncogene, ERBB2, where we actually correlated the number of spots that we got by in situ hybridization with the results that you get from molecular studies such as Southern analysis. And at least for these cell lines, over a dynamic range of about a factor of 10, you get a good linear relationship between in situ hybridization and Southern analysis. So it gives us confidence that this is telling us the true story about what's going on in the tumors. And so this then, from a prognostication point of view, opens the way for 2 kinds of improvements in prognostication. One of them is that we can now rather accurately quantify the number of copies of genes of the amplified genes in these tumors. Even if you have a large number of normal cells intermixed with it, this occurrence would confuse Southern analyses because the normal cells would dilute out the amplified sequence. But with in situ, you can ignore the normal cells, and so you can get a good quantitative measure of the average copy number. This happens to be the average number of CMYC copies in a variety of primary ovarian tumors. So that's one possibility for improving prognostication. But I think the other and more interesting possibility is that not only do we get the mean, but we get the dynamic range, or the variation in the amplification. And what you can see down here, if you just focus on these tumors with the arrows above them, is that if you just looked at the mean, you would really not consider these to be amplified with CMYC at all. But you can see over here that there are subpopulations, sometimes only a few percent, that actually have substantial amplification of CMYC. And so this kind of technology opens the way for prognostication according to the most advanced subpopulation, not just the average. And so this is really a new way of looking at some of the genetic events in these tumors. Now, the other thing that we've been focusing on is detection of deletions. I mentioned that the tumor suppressor genes are typically activated by the loss of a normal allele, and one of the events that can cause the loss of such a normal allele is a physical deletion. And the idea here is to put a probe on the region of the genome that you're interested in studying, put a probe someplace else to use for normalization, and ask the question, can you see the loss of the one normal allele? So what you would look for in interphase is a reduction in the ratio of the number of probes at the test locus to the number of reference probes. This is an example of the application of this in metaphase. This is an ovarian cancer, short-term culture of an ovarian cancer, and we're looking here at chromosome 3. We're using a probe for the centromere of chromosome 3 and also a probe for the mid-short arm of chromosome 3. And what you can see here is that we have a normal chromosome here. Here you can see we've lost the entire short arm of chromosome 3, and we've lost that probe that goes along with it. This would be fairly easy to pick up even using conventional cytogenetics. What's important is that we can also pick it up in interphase without culturing the cells at all. This is the same tumor, and what you can see now is that we have 4 copies of the centromere of chromosome 3, so we have only 2 copies of the short arm of chromosome 3, indicating that there has been a loss of some of the short arm of chromosome 3 in this tumor. What's interesting is that it seems that there are 2 events that have happened in regard to chromosome 3. One of them is this deletion that I was just talking about, and the other is the fact that the tumor became aneuploid, that is to say it doubled the number of copies of chromosome 3. You can see that the structural event occurred before the duplication event, so that we duplicated these 2 chromosomes, and we duplicated those 2. Just to continue that story, we asked the question, does this kind of scheme for detecting deletions really correlate with other molecular genetic events that seem to indicate loss of it, or indicate the activation of a mutant tumor suppressor gene? And so we looked at the gene for p53, and we asked the question, if we look at cells that are normal for p53, haven't lost the normal allele, what does the ratio of the number of copies of the centromere to the number of copies of p53 look like? And what you can see here is that we have about the same number of copies of p53, the black bars, as we do for the number of copies of the centromere, the white bars. So this would suggest that there is no deletion leading to loss of heterozygosity and suppressor gene activation. On the other hand, if you look at tumors that do show loss of heterozygosity for p53, now you can see that we have far fewer copies of the p53 probe than we do the centromere probe. So again, it supports the notion that you can use molecular cytogenetics to pick up events leading to tumor suppressor gene activation. And in this case, the events are physical deletions. Well, the other thing that we've been interested in are mechanisms of gene amplification. And these are biologically interesting, just to give us information about how amplification occurs, and they're also prognostically or therapeutically interesting, as I'll say in a minute. And there are several endpoints that one might be interested in. The first is, how does amplification occur? Is it within chromosomes, or is it external to chromosomes, for example, as double minutes? Do you see co-amplification with other DNA sequences? And what about the heterogeneity within a single cell? All of these things can be approached using molecular cytogenetics. The first question that we asked then was, can we use this technology to tell how the amplification has occurred? This is an example of amplification of the oncogene c-MYC in a colon carcinoma cell line, and in this case the amplification was intrachromosomal. In fact, the amplification occurred predominantly on one chromosome. Here, this is referred to cytogenetically as the homogeneous staining region, or HSR, and you can see that this region shows up not nicely as a chromosomal event, not only in metaphase, but also you can see that it's tightly localized in interphase, suggesting that even if you can't get the cells into mitosis, you can tell that this was an intrachromosomal event just by the compactness of the hybridization domain. If you look at amplification that's occurred extrachromosomally, as double minutes, shown here in metaphase, you can also see that you have sort of a characteristic signature for this in interphase, also more of a shotgun pattern. And so I think that this argues that you can determine, or you can distinguish between intrachromosomal and extrachromosomal amplification, looking just at the interphase nuclei. This is important, I think, from a therapeutic point of view because of the work of Jeff Wall and others, suggesting that if you have amplified sequences that are occurring extrachromosomally, these can sometimes be purged chemotherapeutically, and this may be therapeutically beneficial. In fact, this is a test of that is underway in his laboratory, as I understand it right now. In any case, this technology allows you to diagnose based on the mechanism of amplification. The other thing that's, I think, becoming increasingly interesting is the fact that we can get information about coordinate events. This is an example of a breast cancer cell line that we were looking at with the probes for two different oncogenes. One of them, in red here, was the ERBB2 oncogene, and the other, in green, was the c-MYC oncogene. And what's really interesting about this particular cell line is that you can see that the ERBB2 and the c-MYC are co-localized together in several different places, here, here, and here, but not everywhere. Here's a amplification of ERBB2 by itself. Now, the reason that this is extraordinary is that MYC and ERB are not normally together in the genome. ERBB2 is located on chromosome 17, and c-MYC is located on chromosome 8, and this is neither 17 nor 8, at least the rest of the DNA on it is neither 17 nor 8. So somehow these two sequences have had to come out of their normal site, be colligated together, and then reintegrated back into the genome elsewhere. And this raises the question of whether or not these co-amplification events are somehow important in the evolution of the disease. It turns out that this is, we originally thought, just an isolated event. As we've looked at more and more tumors, we're beginning to feel that this co-amplification of normally separated DNA sequences is perhaps the rule rather than the exception. And so it, again, raises the issue of what role this plays in the evolution of the tumor. Well, once we have developed the capability of studying the known oncogenes, we've become interested in positional cloning. That is to say, in trying to clone other regions of the genome that may be involved in tumor progression. And we now know that there are a number of regions that are commonly amplified for which we don't know the operative gene. There's a region on the 20q-TER in breast cancer that is commonly amplified. There are other regions as well. And there are also regions of common deletion that may be associated with tumor suppressor genes. For example, 16q22 in breast cancer, 9q in bladder cancer, and others. And so another important application of molecular cytogenetics is in trying to define these regions of common amplification or deletion. And the approach that we're taking here is to use interphase and situ hybridization to map out regions of deletion. Here, for example, we're looking at chromosome 16, trying to map out deletions there. And so the approach or the strategy is to use a probe for the centromere and then test probes located at different spots along the short arm of chromosome 16. And we can look in interphase nuclei to find out whether or not you have an equal number of centromeric and test probes, or whether you have a reduced number. If you have a reduced number, such as you do here, then you have a deletion. If you have the same number, then you don't have a deletion. And so what this allows you to do is to look at a fairly large number of tumors and to map out the regions of common deletion. The reason that in situ hybridization is important here is that it can be applied to archived material that's found in pathological laboratories and paraffin blocks. And all of the probes are informative. So if you have a tumor that has a deletion that, for example, involves infrequently lost region, you can map it out very precisely using this technology. And the thing that makes this go is the work that's coming out of the Human Genome Project, more or less worldwide these days. That is, we have a very large number of probes that are distributed all up and down the chromosomes that can be used for this kind of mapping. And so I think that this approach to genetic analysis is going to become increasingly important and powerful as the Human Genome Project progresses and generates more probes for us that can be used in these kinds of studies. Now, what I've talked about so far is fairly routine fluorescence in situ hybridization. The power of the technology is that it allows you to look at individual cells and see extremely small genetic events and aberrations. The difficulty with it is that you have to know where to look. And so if you're interested in studying solid tumors, which are highly heterogeneous, you have to apply a very large number of probes and a very large number of different hybridizations in order to map out the genetic events that are going on in it. And so what I want to talk about in the last few minutes of this presentation is a new technology that we've developed in the lab in San Francisco over the last 6 months or so called comparative genomic hybridization. And this does not have that limitation. This allows you to look at the entire genome in one experiment. And the way this works is to turn it around. Normally you hybridize with a probe the cell line that you want to look at. Here, you take the tumor DNA, for example, and you use that as the hybridization probe to a normal metaphase spread. At the same time, you hybridize competitively with normal DNA. And you do this so that the tumor DNA can be detected in one color and the normal DNA can be detected in a different color, let's say red for normal and green for the tumor. And you then use the image analysis system to measure for each normal chromosome the red-green ratio along that chromosome. Now, if there's a region in the tumor genome that's amplified, you'll have a higher copy number of the tumor sequences in that region than you do normal, and so you would have a high green to red ratio, and you get a peak here, and that would tell you that you've got an amplification event, and it would tell you on which chromosome and where along that chromosome it occurs. If you have a deletion in the tumor DNA, then you'll have a low green to red ratio, a dip here, and again, that will map out that region of deletion. And so the advantage of this is that in one hybridization experiment, you can map out most of the regions of amplification and deletion in that entire tumor. And this is an example of how that works. This is application of this technique to the cell line, the colon carcinoma cell line. This is just off the image analysis system that shows you where the chromosomes are. This is the green image. And so this is hybridization with the tumor DNA. This happens to be chromosome 8, and what you can see out here along the end of chromosome 8 is a bright green spot. If you look at the same chromosome in the red domain, which is our normal probe, what you can see is just more or less a uniform distribution of red fluorescence along it. And so if you actually quantitatively measure the fluorescence profile along those chromosomes, you see a peak in the green fluorescence, you see nothing in the red fluorescence, and so if you look at the red-green or the green-red ratio, you can see a peak out here at chromosome 8q24. It turns out that we knew in advance that this cell line carried amplified c-MYC. In fact, I showed examples of that earlier using a specific probe. What you can see here is that we've detected the presence of that amplified oncogene without knowing what it was. We do know that since it maps to 8q24, that it's most likely c-MYC. But that's not particularly interesting if you know what's amplified in advance, but if you don't, then it becomes much more interesting. This is a breast cancer cell line, SKBR3, which is known to be amplified also for c-MYC. Again, this is chromosome 8 here, same representation as before, and you can see out at the end of chromosome 8 there's a bright green spot here corresponding to c-MYC. What we didn't know was that there's another region on chromosome 8 that's also amplified, and if you look at the profiles here, you can clearly see these two regions of amplification, and you can map them. So it's a very powerful technique for detecting all of these amplified regions in the genome in one shot. It also allows you to detect duplications and deletions. This is another breast cancer cell line. Here we're looking at chromosome 1, chromosome 11, and chromosome 16. Here you can see that perhaps you have too much on one part of chromosome 1 and too little on parts of chromosome 11 and 16. In fact, you can use the image analysis system to trace out these changes. Here you can see a trace along chromosome 1. You have too many copy or too much signal on chromosome 1q, about the normal amount on chromosome 1p. So this says that we've got a duplication on chromosome 1q. If you look at chromosome 9, we've got a deletion on 9p, we've got a deletion on 16, we've got a deletion on 11q, and we've got a deletion on 17. So in one experiment, we've mapped out all of these deletions and the amplifications. And so what you get, then, is this very powerful technique, which we're calling comparative genomic hybridization, that allows you to look at or to generate what amounts to a chromosome copy number karyotype that shows you throughout the entire tumor genome what's amplified and what's deleted. And we think that this is going to have real importance in trying to identify some of the additional genes that are associated with the progression of these tumors. Skip that. If I could have the lights. We've been applying comparative genomic hybridization for some time now, and I think it's fair to say that about 80% of the sequences that are amplified or deleted in these various cancers so far can't be associated with any known genetic aberrations. I think it's going to certainly give us some important clues as to how these tumors evolve. Well, let me just summarize by saying that fluorescence in situ hybridization is really quite well developed now. It allows visualization of specific sequences both in metaphase and interphase. It's useful in physical mapping and investigation of the 3D organization of the interphase nucleus, as you'll see later in the conference. And it's certainly proving to be very powerful for genetic characterization of these diseases. And I think that we're also excited about the fact that this comparative genomic hybridization complements it by allowing us to pick up new regions of amplification, even if we don't have probes for them. Eventually, the idea will be to convert those regions with specific probes so that we can apply fluorescence in situ hybridization even on single cells and detect those events as well. Thank you.
Division for the light heavyweight.